Hello, everyone, and welcome to the Sequence 2 assembly session. I'm Fergal Martin. I'm the coordinator for vertebrate annotation at Ensemble at EMBL EBI. And with me is my co-chair, Nancy Holroyd, who leads the Tree of Life sample management team at the Wellcome Sanger Institute. So this kind of follows on from the specimen sequence session yesterday and is kind of part of a trilogy with the assembly to genome session concluding it tomorrow. And it gives you a kind of an overview of everything from sampling stuff in the field right up to sequencing and assembly and ultimately annotation and downstream analysis. So we're lucky today to have 5 talks that are exciting kind of new works in the field. We're going to start with Claudia Weber from the Wellcome Sanger Institute, who's going to talk to us about methods for separating cobionts and contaminants from DTOL samples. Then we'll have a kind of a double header talk between Jean Myers of the Max Planck Institute of Molecular Cell Biology and Genetics, who's going to talk about K-mer counting via FAST-K. And we'll have Yoshihiko Suzuki of the Okinawa Institute of Science and Technology talking about K-mer classification using ClassPro. And then we'll have Giulio Frumenti from the Rockefeller University talking about improved variant filtering and polishing via K-mer validation using a tool called Murfin. And we'll wrap up with Alan Tracy from the Wellcome Sanger Institute talking about issues in terms of purging haplotypes from apple assemblies. So just in terms of the layout of the session, all the talks are going to be played back to back, and then we'll have a Q&A session with the speakers afterwards. So please put any questions you have for the speakers into the chat box. Do tag speakers you want to direct the questions at, although more general questions are also okay. And ideally put them in before the start of the Q&A session so we have a chance to see them. And do upvote any questions that you'd like to see answered. So without further ado, let's start with the first talk from Claudia.
Hi everyone, today I'll be talking about disentangling sequences from different sources in Darwin Tree of Life samples. So when we go to sequence a sample, in addition to the target species genome, we'll often find that there are some contaminants in there and sequences from cobionts. So we would like to get rid of the contaminants, but generate a separate assembly for the cobionts, because there are some biologically interesting things that can be learned from that. For example, we've seen some moths that have some Wolbachia endosymbionts in them, as well as some vertebrates and some parasites that interact with them. In the current Tree of Life pipeline, there are contamination checks that happen at the end when the genome has already been scaffolded. And these are largely based on BLAST with some additional steps if needed. But what we'd like to do is make this process a bit more speedy and efficient. So we want to cast a slightly broader net and start binning things by their probable origin, starting from the reads and the contigs. And we'd also like to reduce our reliance on databases in the process. Why are databases a problem? Well, with some things that we sequence, there might not be any close relatives currently in databases. And this will be particularly true when we get to sparsely sampled parts of the Tree of Life. So it can be hard to assign where a sequence came from. In addition, the databases can also be contaminated. So we may end up either having no hits to a sequence that we're trying to assign or an incorrect or inconsistent assignment, and therefore we might not pick out the contamination accurately. So what we can try to do instead is separate the problem into 2 parts. The first being working out which sequences belong to the same group of things, and then asking separately whether we can find some reliable information about where they might have come from. So our sequence binning process, which we call cobiont identification, has got 2 main parts. The first is to make a rough taxonomic assignment based on markers, and then narrow that down a bit. And because this is still database guided, we can also look at the sequence composition. Now, the intuition behind this is that microbes and animals will have different tetranucleotide compositions, GC contents, coding densities. So we can use these features to try to cluster those sequences and group them together. In addition, we can look for physical connections between molecules to work out whether something came from the same genome or not. The marker scan pipeline, which was set up by my colleague Emmeline, works by taking the contigs and applying a hidden Markov model to them. And that pulls up the ribosomal RNAs, which are quite conserved, so they've been quite well sequenced. And those are then used to get an approximation of which taxa are in the mix. This information is then used to make a custom Kraken database that allows us then to classify the reads and split the target, say the moth, away from its covariant. And we then take the possible covariant reads and map them back against the assembly. And if we find contigs that are well covered by the reads, we can say that those probably came from a covariant. So we also confirm that the reads belong to the covariant rather than, for example, being spurious hits along the sequence or some stretches of horizontal gene transfer. And then we've got another step wherein the flagged reads are also independently reassembled. Now, in order to bin by sequence composition, what I do is for each sequence, tally up a set of statistics. And this is primarily using the tetranucleotide composition of the sequence. So I end up with a big feature matrix for my set of sequences. And then to get that into 2 dimensions so I can visualize it, I put it into a variational autoencoder. When I apply this to the contigs of the buff-tip moth, and then label those contigs by the estimated coding density, we can see that the Wolbachia form a distinct type cluster and they stick out because their genomes are denser than that of the moth. You can also apply the same technique to the read sets. So the first panel again shows the buff-tip moth. And here I've pulled in the labeling that came from the marker scan. And as you can see, there's a distinct cluster that's been tagged as Wolbachia by the marker scan. So this one's relatively straightforward. The second example is a bit more interesting because we start getting into the issues of having database gaps here. So the London dowd, which is another moth, also has a bacterial infection. So there's a cluster of Erwinia at the bottom. And at the top, there's a cluster that's sort of half labeled as coming from Microsporidium, but it looks as though half of these reads have been missed. So that suggests that maybe we're just missing something that's close enough in the database. And we were able to confirm this by performing a mapping step, which shows us that this all actually belongs to the same entity. So we can make this process robust to those database gaps. So the grand scheme of things, this is still relatively straightforward because we're looking at a microbe that's infecting an insect. But what happens if we've got 2 animals that are in the mix? So the problem with animal genomes is that they're overall less well conserved. So the chances of assigning something based on the database are going to be lower. And in the case of the Allophone tuna, that's exactly what we saw. So the traditional scan didn't pick up anything obvious here. But if we look at the composition of this genome, we can see that there are some large fish scaffolds in the center of the plot. And then there's another group of scaffolds that either have no hits or inconsistent spurious hits. So it's slightly unclear where these came from. But when we ran the marker scan, we were able to detect some ribosomal RNAs from a myxozoan parasite. So the question is whether all of this belongs to the parasite or not. And here we can look at physical interactions between the molecules. And when we look at the Hi-C map, we can see that the unassigned cluster does not interact with the rest of the fish genome, so it appears to be separate. Now, getting that Hi-C map into the state is quite a bit of work. So there's a human who has to sort all of these things, and we wondered if we could speed this up somehow. So what I've done here is constructed a matrix of all of the pairwise interactions that were found in the Hi-C data between the scaffolds. And each quadrant that's labeled white has at least one connection. I then turn this into a network graph. And what we can see here is that this network splits into about 3 different pieces, with the top one containing the parasite. And in order to be able to extract these efficiently, I generated a set of embeddings, which gives you a measure of how similar these things are to each other, and use that to label them, which the colors that you can see in this plot here. When I now resort the matrix based on these labels, we can see it's a lot tidier, and there's definitely something that stands out here. More importantly, we found that the network clusters are also consistent with manual assignments that were made by curators, and there's an overlap with the composition cluster as well, which suggests that adding this information is suitable to help remove cobalance. So in summary, we've seen that markers can be useful to approximately taxonomically place sequences, even when they become quite diverged. Composition can help separate sequences, even in the absence of other information, so we can use that as a useful backup. And if we combine markers, composition, and network connectivity, we can potentially accelerate the whole assembly decontamination process. So at the moment, this process is being integrated into Sanger's Tree of Life pipeline, as well as the ToolQC pages, which provide a nice overview for all the species that have been sequenced. And we'll be assessing how this impacts the throughput through the pipeline and adapting it to handle more tricky samples. For example, we'll be having samples from the aquatic symbiosis genomes coming in soon. And with that, I'd like to thank all of the people who contributed to this work and provided comments, particularly Emmeline, who did all of the marker scan work. And I'd like to thank you all for listening.
Hi, I'm Gene Myers, here with Yoshihiko Suzuki from the Okinawa Institute of Science and Technology. And today we're going to talk to you, we're going to give a combined talk about our work on K-mer counting and what we call K-mer classification and their application to genome assembly and analysis. FastK is a K-mer counter. That is, if you give it a sequence data set, it will count the number of occurrences of each distinct K-mer in the data set. It is optimized for high accuracy shotgun data sets, and it is a refinement of the disk-based minimizer partitioning algorithm found in the KMC3 K-mer counter. It can produce 3 different outputs. First, a histogram of the frequencies of the K-mers. Second, an alphabetically sorted table of the K-mers found in the data set, along with their counts. And finally, a read profile for each read that gives the count of each consecutive K-mer along the read. A profile is not a new concept per se, but FastK is the first K-mer counter to efficiently produce profiles as an integrated component of its overall function. In the profile shown, one can clearly see the K-mers that are diploid or haploid in the underlying genome or errors in the read. And we'll talk about this a bit more later. Some of the features of FastK are as follows. There is no limit on K, the K-mer size. It uses 16 gigabytes of memory, regardless of the input problem size. It supports all the standard input sequence formats. It is threadable up to 128 threads. And lastly, it can produce something called a relative profile that gives the counts along a read with respect to another data set. And these are particularly useful for applications such as trio binning and polishing. As an illustration of performance, consider a 50x hi-fi data set for Cabernet Sauvignon that consists of 28 gigabytes of data. Shown are the calls to KMC3 and to FastK with the correspondence between the parameters indicated by the arrows. So FastK, if you look at the times, FastK produces the histogram and table about 1.65 times faster, and the table occupies 17% less space on this particular problem instance. And if you additionally set the -P option, FastK will take another 3 minutes to produce a compressed representation of all the profiles that occupies about 16 gigabytes. This 3 minutes is for an inverse sort of the k-mers, and it takes much less time than it would take to produce the profiles post hoc by looking up the k-mers in the table. And with the compression, the profile index occupies only about 0.56 bytes per base in the data set. In general, what we find is that FastK outperforms all previous k-mer counters for shotgun data sets of 20x or more, and with an average read error of 1% or less. Okay, so here's a popular list of genome analysis tools that use k-mer counters. And here are the 3 k-mer counter programs they depend on. What's important is that none of the outputs of these programs are compatible. Therefore, if you want to run all the tools, then you need to install all 3 counters. And if you want to keep the tables around, then you have to keep 3 versions of the table on your disk. Shane McCarthy at the Tree of Life project suggested that it would really be better and it would be great if all these tools operated with respect to a single k-mer counter program. And so we've embarked on doing that for FastK, and we've almost completed the task. In doing so, we ended up replacing smudgeplot with a new program that we call ploidyplot that produces much more interpretable plots. And we only realized the tools in CAT that are not in Mercury, as these 2 tool suites are quite similar. We also have realized a hi-fi shotgun simulator called HiSim that models arbitrary ploidy and uses an error model that is derived statistically from analyzing a hi-fi data set. And in a few moments, Yoshihiko will show you our ClassPro application that accurately classifies k-mers using read profiles. At this time, the only components missing are the trio analyses of Mercury. Everything else is available in repositories at my GitHub account. When we developed a threaded incremental version of smudgeplot, we suspected that many of the smudges that we were looking at were artifacts. They just didn't make sense. For example, we demonstrate this here with a simulated 50x triploid data set with 1% heterogeneity between the haplotypes. This data set, incidentally, was produced by HiSim. In the smudgeplot output, you see the expected triploid smudge, but also circled in red is a much brighter smudge that should occur really, truly only in diploid genomes. This smudge, after analysis, we found is due to artifactual counts produced when head sites are within K base pairs of each other. So in ploidyplot, we developed a method that detects this situation, then doesn't count the hapmer pairs from this situation, giving a clean single triploid smudge as you see there. So all of the applications that we've been discussing were implemented from scratch, often with the best possible algorithms for each task. Because I re-implemented or started anew on all of these, all of the programs are threaded, can be threaded. They all efficiently stream the tables, and so you don't need a high mem machine to basically run any of the programs using FastK. They all share common conventions and options. For example, Ronri in the Mercury suite has a nice fill, line, and stack display option for her histograms. I basically took that same idea, and we also use it for ploidyplot to indicate heat map, contour, or stack of both. So finally, let me turn to the k-mer classification problem. Given the k-mers and their counts in a shotgun data set, what we would like to know truly is how many times each KMER occurs in the underlying genome. In other words, does it occur twice for a KMER occurring once in each haplotype of a diploid genome? It occurs once for a KMER occurring in only one haplotype, or it would not occur, it would occur 0 times for a KMER that contains a read error. We call this the classification problem, and Yoshihiko will now present a novel and accurate method using read profile.
Hi everyone, I'm Yoshihiko Suzuki. So here I introduce a new KMER classification method based on the read profiles generated with the FastK program. So this is an example of the profile of a single HiFi read in a simulation dataset with 40x coverage. In this plot, each dot indicates the count of the KMER ending at the position on the read, and adjacent KMERs are connected with gray lines. And the color of the dots indicates the count of true classification of each KMER, which means the copy number of the KMER in the underlying genome. And the KMER counts are capped at 70 here, but for example, the counts in this region are actually about 1,000. And the almost only approach currently used for the KMER classification problem is to use the global KMER count histogram. In this histogram-based classification, we first calculate the count thresholds between the different classes based on the histogram, and then simply use those thresholds to estimate the KMER class, like this. However, as you can see from the orange marks that mean wrong classification, this histogram-based approach tends to fail, especially around the boundaries of the thresholds and also within the repeats. So how can we leverage the read profiles for more accurate KMER classification? Here I introduce some key principles to understand the transition of the KMER counts in a read profile. So the first principle is coherence principle, which means adjacent KMERs with the same class have similar counts. The fluctuation of the counts between these KMERs having the same class is due to a Poisson process of read start and read exit, and also sequencing errors in reads other than the subject read. But the important thing is that these effects are both very small, especially in HiFi reads. And the second principle is K-knockout principle, which means transition from a higher class to a lower class lasts for approximately K bases. For example, single sequencing error or heterozygous variant affect approximately K consecutive KMERs. And the last thing is that the fact that in HiFi reads, there is also some complication due to higher error rates of low complexity sequences. For example, a homopolymer sequence of 10A has an error rate higher than 10% in HiFi reads. And now I move on to sketching the algorithm. So given a read profile, we first detect the segments in each of which the KMER class is identical using the coherence principle in the previous slide, while considering the complication due to low complexity sequences. For example, we can infer that the count drop here is not because of a class change, but because of homopolymer errors in other reads, according to the homopolymer length shown in the blue histogram at the bottom. And if a position is not explained by the coherence principle, then that position is called a wall, where the KMER class changes. And the intervals consisting of erroneous KMERs are also identified here using the K-knockout principle. So starting from all the KMERs, we identify the walls where we can actually correct the counts at the walls for long intervals, which clarifies the curves that would consist of haploid KMERs and also diploid KMERs like this. And now it looks almost obvious, but we classify these long intervals using a dynamic programming-based method to find the haplotier and diplotier. And using these haplo and diplotiers, we finally classify the rest of the k-mers while considering the variable error rates depending on the sequence context, like homopolymers. And as for the performance on the simulation dataset, ClassPro showed 99.4% accuracy, whereas the classification based on the global k-mer count histogram was 95.6% accurate. And because we can't calculate the accuracy for a real dataset because we don't know the truth, but real datasets usually have a much larger read sampling fluctuation, which means the histogram-based classification becomes even harder, although ClassPro still can, in principle, classify such reads accurately using the context information. And the overall accuracy is 99.4%, and there is still 6.6% error, but the conditional accuracy only for the non-repetitive reads is 99.9%, which is almost perfect. So yes, the accuracy gets weaker for repetitive reads because the information available in the read profile decreases. But one advantageous point of ClassPro is that the false negative rate of erroneous k-mers is very low, where the histogram-based method fails to detect 1.5% of the erroneous k-mers, but ClassPro does only 0.5%. Actually, ClassPro's classification for the same k-mer can be inconsistent among different reads because ClassPro classifies each read independently, although the overall accuracy, consistency of the classification is 99.9%. So we will offer an option to generate the consensus of the classifications for each k-mer in a dataset. So the beta version of the ClassPro program is available here, and it is still under active development. Thank you very much.
So we're excited about ClassPro because having accurate classifications enables more accurate and often more efficient methods for downstream genomic applications. For example, simply plotting the histograms for the 4 classifications immediately gives a genome scope decomposition. Knowing the haplotypic specific k-mers gives more specific read mapping, and being able to avoid k-mers, error k-mers, allows one to use fewer seeds for the mapping, giving greater speed. For trio binning, one needs to only focus on the head sites within each child read. And for spectral alignment, it's simply a matter of avoiding the paths with error k-mers, which we know quite reliably. And finally, as for the read mapping, assembly overlap can be faster and phase sensitive. So we'd like to thank the following individuals for their valuable feedback, suggestions, and beta testing over the course of our work over the last couple of years. Thank you very much.
Good day, everyone. Today I'm presenting Murfin, a tool for improved variant filtering and polishing via k-mer validation. This has been developed in collaboration with the Telomere to Telomere Consortium and the VGPE project. The motivation behind the development of Murfin is that assemblies contain base calling errors. These errors can affect downstream analysis, such as gene annotation, and this issue is exacerbated in noisy long read-based assemblies. This is why polishing has become common practice. However, no single set of criteria can be defined on how to filter the variants used for polishing. Murfin, on one hand, can be used to evaluate the consensus sequence, and on the other hand, it can be used to improve it by filtering variant codes. The basic principle is that we're aiming to reduce the number of false positives in our variant code set, while not significantly affect the true positives. This should lead to better precision while not impacting the code. The way we achieve this in Murfin is based on a number of relatively sophisticated algorithms and statistical approaches, some of which are based on a revised version of GenomeScope 2 statistical models, which allow us to predict essentially the k-mer expected frequency So we can then decide whether these are errors or they are found at expected frequency in the assembly. This can be used to evaluate the assembly by computing a series of metrics, or it can be used directly for polishing by deciding whether a variant is going to negatively have an impact on the assembly or not. Here you can see an example. The Murfin was initially developed to polish and evaluate SAGE-M13, the new human genome assembly, which is complete and accurate. And here you can see that one of the metrics defined within Murfin, K-star, can be used to evaluate the quality of different versions of the assembly. So the V1 version here in blue can be compared, for instance, to a standard assembly using HiCanoe. And you can see that the K-star in that case should be distributed around 0, and the narrower the distribution, the better. You can also use the K-star to evaluate individual k-mers. And here you can see, for instance, that in one of the earlier versions of the assembly, which was not yet complete, there are some k-mers that show here in the upper right quadrant, meaning that they are actually underrepresented and missing from the assembly. You can also evaluate the base level accuracy of the assembly. So here you can see the K-star using HiFi k-mers and Illumina k-mers for all the chromosomes in SAGE-M13. And generally, you should expect this to be around 0, but you can see that deviations from that are useful and can potentially be further investigated. So for instance, here you can see that there is a region here on chromosome 9 where there are significant deviations both in HiFi and Illumina k-mers. And this is because of the inherent bias in terms of sequencing of these technologies when it comes to repetitive elements such as HSAT repeats. However, generally speaking, throughout the assembly, and particularly within outside repetitive elements, you would expect a K-star around 0, as you can see here, which is also supported by the Reed PyLab. However, in other cases like here, you can actually see that whenever there is a potential issue, this is also reflected within the K-star. Here is another example. This is the known bias with the GC or GA reach repeats. And here in this case is the GA repeats that HiFi reads tend to have a problem with. So you can see that essentially HiFi reads here have a dropout in the coverage, which is also reflected in the K-star. And this is actually happening in a similar way with the PCR-free data sets. ONT seems to be better in this case. We then tested and used Murfin to polish and evaluate a genome assembly from the HZ02 data sets. In particular, this is a trio data set, which we assembled using nanopore reads. And here you can see that we are improving the consensus accuracy, the blue distributions over the red ones, when we apply Murfin to filter the variant calls generated by Medaka. This is true for the vast majority of contigs within each of the assemblies. Importantly, this is happening despite the fact that we're not significantly affecting phasing. Actually, we're maybe improving it. As you can see here, the blue lines, again, they are comparable to the Medaka-only version of the assemblies. And very importantly, we're doing this without introducing much fewer variants. So using much fewer corrections in the assembly. Again, here the blue boxes compared to the red ones. So it means that we're actually introducing much fewer changes in the assembly. And despite that, we're achieving better QV and comparable, if not better, phasing. This, of course, is reflected by generally higher F1 precision and recall scores, particularly when it comes to eDELs. We also tested Murfin within the VGP pipeline and then introduced it starting from version 1.7. Here you can see at the polishing steps, we are now adding Murfin. And again, we observe that this is basically improving the quality of the consensus accuracy for all the assemblies as judged using Mercury QV, but also when it comes to gene annotation, these are results using the NCBI annotation pipeline. So we observe a significant reduction in terms of the number of premature stop codons, frame shifts, and finally low quality proteins within the final results. We also observe that Murfin can actually be used not just for polishing, so not just for filtering variant set, use it for polishing, but actually can be used for filtering any variant set. For instance, from a population resequencing data set. And this will lead to a better precision by reducing the number of false positives. Again. In conclusion, Murfin computes several assembly metrics that can be used to infer the quality of assembled genomes or variant sets. And then, using these metrics, Murfin can significantly improve polishing of genome assemblies and variant filtering in general. I would like to acknowledge all my colleagues who helped and contribute to this, and in particular, Aaron Reed. Murfin is available in GitHub, and we also submitted a paper for consideration, which is already available as a preprint on Bioarchive. So we encourage you to test Murfin and also to read our paper for additional details. Thank you very much.
Hi, my name's Alan Tracy and I work in the Genome Reference Informatics team at Sanger. The work I want to describe today has been carried out by myself and Shenxi Jiao from the Tree of Life assembly team at Sanger. During our work on assembly and curation, we see many different genome assemblies from all manner of different species. However, we've done very few polyploid genomes and very few plant genomes, so we know that there are bound to be some new challenges awaiting us as more assemblies come through to curation. When we looked closely at the first apple assemblies, we noticed something interesting that we hadn't seen previously. That's what I'm going to talk to you about today. Here, looking at the first apple assembly to come into curation, DR Meldome 11, this is a plot of our chromosomes aligned to the best existing apple reference. The two assemblies are very nicely collinear, which we would expect, apart from, most noticeably, one large region on chromosome 13. When we zoom in on this, we see a region of around 7 megabases that are largely absent from our assembly relative to the reference in this region of chromosome 13. Just in case a sequence was lost in the purging process, I checked our assembly against the pre-purged assembly, and they both show the same amount of missing sequence. Okay, so here's a comparison of chromosome 13 in DR Meldome 11, the one I've been showing you on the bottom, versus another Sanger assembly, DR Meldome 10. This time, at the same locus, there is 3.75 megabases missing from DR Meldome 11. So from a comparison with two different genomes, DR Meldome 11 is missing at least 3.75 megabases. When we take the missing sequence that's present in the other Sanger assembly, DR Meldome 10, and probe the DR Meldome 11 alt assembly, we find that the sequence is not only present, but the whole thing is present twice. We know from the names of these two sequences, the ATG prefix, that they were removed erroneously by Hi-Phi-asm. Hi-Phi-asm incorrectly classifying both of these sequences as haplotypes, leaving a large hole in the primary assembly. When we bring one of these two contigs back into the primary, ATG32L, we find that it sits very nicely in the gap where we expected to have a missing sequence based on comparison with the reference. So with a little investigation, the problem has been able to be manually fixed. Looking at another Sanger apple assembly, DR Meldome 5, we see the same chromosome, even the same locus, but not just 3.5 megabases. This time, the last third of our chromosome is missing. That's 15 megabases of the reference is not present in DR Meldome 5. And when we look in the alt assembly, we see the whole sequence present, this time just once, totaling just over 12 megabases. The contig name prefix is PTG, indicating that this large sequence was incorrectly removed by purge dupes. So I've shown you two examples of large sequences representing significant chunks of the genome being incorrectly removed. And this is not happening in just one software tool. This is two separate tools. Hi-Phi-asm and purge dupes have both incorrectly removed chunks of our primary assembly as haplotigs. Focusing on DR Meldome 5, the case where purge dupes incorrectly removed 12 megabases, this plot shows us the sequence alignment between the removed contig and the contig that purge dupes deemed to be a haplotype of that contig. we can clearly see that there's no collinearity between these two sequences. And in fact, PERJ-dupes doesn't require collinearity. In this case, the haplotype has been called because 70% of the length of the contigs are in common with each other. So 70% of one contig is found in the other. This rule has not been a problem for our other genomes, but clearly there must be very large amounts of sequence genuinely in common between different regions of the apple genome. And here we have another case from a third Sanger apple, DR Maldome 58. Again, another case of PERJ-dupes incorrectly calling a haplotype and removing it from the primary assembly, around the same locus on chromosome 13. When we compare the removed contig against the contig it was supposedly haplotypic to, we can see that in this case partial collinearity does exist between the two contigs. This collinear section plus lots of smaller matches were enough to push similarity up above the 70% threshold for incorrect designation as a haplotype and subsequent removal. Okay, so summarising what we found, we can see at the top 3 cases of incorrect purging by hifiasm in 2 different apple assemblies, and at the bottom 3 cases in 2 different assemblies erroneously purged by PERJ-dupes. As I've already mentioned, the same region of chromosome 13 suffers the deletion in all 3 assemblies to a greater or lesser extent. DR Maldome 58 is somewhat different to the other 2 assemblies, in that not only was sequence missing, a lot of retained duplication or failed purging was also found, much more than in the other 2 assemblies. Here's a Hi-C map of part of the assembly. You can see from the strong off-diagonal collinear matches, which incidentally coincide with half normal coverage, that we have a lot of retained haplotypic duplication in this assembly, which needs removing, but which PERJ-dupes has failed to detect and deal with. When we look at a smudge plot of this assembly, produced from Gene's ploidy-plot summary tool using Mercury FastK, we can see the main peak at 3n, suggestive of the assembly being triploid. To further confound the task of removing haplotypic duplication, we see another peak around 6n, suggesting some regions of the genome are hexaploid, which we think is likely due to a recent genome-wide duplication event. PERJ-dupes was written to deal with retained haplotypic duplication in diploid assemblies, and was not intended to be able to deal with polyploid scenarios. So, going forward, we are drawing up plans to better deal with purging in polyploid genomes. We plan to refine the metrics for haplotype identification, think about a check to reinstate overpurged contigs, and think about how we can avoid underpurging for highly divergent haplotypes in polyploid assemblies. That's all for today. Thanks for listening, and we're happy to take your questions.
Welcome back, everybody. Sorry for the short technical glitch that we had earlier after the first talk, but thank you for sticking with us. Thank you very much for the excellent talks from the speakers. It's really good to hear about these advances in assembly technology and expertise. It's great. We have only 4 or 5 minutes for a few questions. So I'm going to kick off with a question for Claudia from Kim Walden. So Kim's asked, we work on small insects which sometimes feed on other insects prior to collection. Do you think your approach would discriminate between 2 genomes within the same phylum?
So that's a really interesting question. I think it depends a bit on how distinct those genomes are and how much of the smaller insect has been eaten. If there's a decent amount, I would expect that the marker scan would pick that out. And then when you start looking at the contigs, I expect those to separate maybe a bit more than the reads. But I put my money on the Hi-C if you're able to get that, I think.
Okay.
Would, tell you if something's physically connected or not.
Yes, okay. Thank you very much. A question for you, Jean, from Kim Walden. Are GeneScope histograms more accurate with FastK data versus jellyfish versus Merrill without ground truth sets?
Actually, the histogram will be exactly the same. for all three of the Kamer counters. They give exactly the same counts. What GenomeScope does is it fits negative binomial distributions to basically estimate the histogram of 0 copy, 1 copy, and 2 copy Kamers, classified Kamers. And what I was saying basically is that after running ClassPro, you know exactly which Kamers are 0, 1, and 2 copy. And if you simply just plot those histograms, those are basically the curves that GenomeScope is trying to produce, and they're better curves.
Okay, thank you very much. I hope that answers your question, Kim. And following on from that, Yoshihiko from Wuhan, does the tool work well in multiploides genomes? Will the intervals be too short to count if the heterozygosity level is too high?
It's currently only for diploid genome, but yeah, we are eager to extend the algorithm to multiploid genome. So the question about the high heterozygosity is, I don't think it matters, the higher heterozygosity matters, because ClassPro works well if the haploid layer is clear. So one allele hetero variant makes an interval of approximately k base pairs in length, and which is sufficient for the definition of the long interval. So it is no problem.
If I may, Yoshihiko, the drops are the same. What happens is you go to ploidy is that there are more tiers to distinguish, and distinguishing the tiers does get harder. But the generalization is clearer, and we do hope to work on it in a later work.
Right.
Thank
yes.
you for letting me know.
Thank you both. Julia, a question for you from Kim Walden. What are your thoughts on Murphin polishing PacBio HiFi genome assemblies? Is it beneficial or is it not?
Thank you for the question. I think it depends what you want to achieve. I mean, HiFi reads were introduced very recently, scientifically speaking. And of course, we're all very much enthusiastic about them because they definitely fixed or improved the consensus accuracy of our assembly. So of course, we're now much happier than we were just a year ago. For instance, when we annotate our genomes, we find less issues with them and so forth. But what I think it's clear already is that even with HiFi reads, assemblies are not perfect. So if you want to achieve a very high quality assembly and with the perfect or near perfect consensus accuracy, as it was, for instance, the case of the CHM13 assembly, the telomere to telomere assembly of the human genome, it's clear that you still need to polish. It still means that after whatever assembly tools you've been running, there will still be errors that needs correction. And Murphin does just that, tries to improve the accuracy of that correction. So for instance, if you look at the polishing paper that was published, it's bioRxiv that hopefully will be published soon for the polishing of CHM13, it's clear that Murphin improves these variant calls aimed at correcting some of these remaining errors even after HiFi assembly. So in that paper, I think we suggest that Raccoon plus Murphin is kind of the best strategy for that.
Okay, thank you, Giulio. I have one more question for Alan. I hope you don't mind us going over time and indulge me a little bit here, just to ask whether you think you'd be able to spot such assembly issues if you didn't have a reference genome to align to.
I think it would obviously be harder, but now that we know that this issue exists, we can we can look to the alt assembly
perhaps more than we have been.
Okay, thank you very much, Alan. Okay, so I think we're a couple of minutes over schedule. We should call it a day here and get ready for a coffee break. I encourage you all to stream D tomorrow, which continues from assembly to genome. Please join us for more excellent talks, and thank you to our speakers. I highly recommend returning at 7 o'clock to see Spellcast in the Tree of Life. They're a group of performers based in Nairobi who recently featured on East Africa's Got Talent, and they'll be treating us to a world exclusive, never before performed set. So thank you very much. Thank you for our speakers, and see you at the concert.